Okay, all right. So everyone can see, all right. Awesome, great, so let's jump in. So welcome everyone to our macroinvertebrate training. So you all are here. You know, maybe you are already familiar with macroinvertebrates, or maybe this is your first time learning about them. But thank you for joining us, and we have a lot to cover in this afternoon. So kind of like how I went on a moment ago about what is Georgia Adopt-A-Stream? What are we? So we are the state's volunteer water quality monitoring program. And like I said, we train and encourage citizens to monitor the water quality of different waterways all around the state. And we have 5 main program goals that we stick to. And these goals, lovely enough, they line up here in a mnemonic device, so adopt. So A being the first goal is increase public awareness. Our program is housed with Georgia's Environmental Protection Division in the Water Protection Branch in the non-point source program. So we really work to raise public awareness about non-point source pollution and its effects. So part of our program goal is to just increase public awareness of non-point source pollution and teach people the different impacts that this has on water quality. D stands for data. So with our program, our volunteers collect quality baseline water data, which just tells the baseline quality of their stream. So you can think of this baseline data almost like as a heartbeat of the stream. And what you're looking for is differences in this heartbeat, and to see those abnormalities and to then kind of pinpoint, all right, what was going on during that time? What is the source, or what is the cause of this abnormality in the data? O stands for observations. So we encourage our volunteers to not only just gather observations about their monitoring site at their waterway, but also take note of the changes that are going on within their watershed, around the land, in their community, because these changes align with you know, non-point source pollution. So you'll be able to understand the land use effects and how that impacts the water quality. P stands for partnerships. We encourage everyone to form a partnership with their local government or with just other community members. Really, it's to help you spread awareness, to help educate others, and really, you know, just expand that outreach effort. But it also, these partnerships will help you maybe obtain equipment, or just expand your monitoring project too with others. And lastly, T stands for tools and training. So our state office, we can help provide, you know, equipment for you when monitoring. There's local coordinators all around the state that you can link up with, and they can help provide some tools for you. Of course, we also provide workshops and, you know, the training to teach you about, you know, water quality and the actions that you can take. So, you know, some of you may already be aware, but we have chemical workshops, bacterial, watershed, visual. It really goes on and on. But that brings us to today's workshop, which is specifically on macroinvertebrate monitoring. So what is it? What does it involve? And just a little FYI, I will probably be saying macros, and macros is going to be referred to on a lot of these slides, and that's just short for macroinvertebrate. So macroinvertebrate monitoring involves the collection, the identification, and the counting of these macros. And the purpose is to just quickly assess both the water quality and the habitat quality of your site, of your waterway. So we do this by taking note of the abundance and the diversity of the macro populations sampled at these sites. So even though we do collect and note the abundance and diversity, our macro key, which we will discuss later on, places a really, it places more importance on the diversity of populations because as we'll learn later on, some macros, just because you have a higher abundance of them, a greater number, doesn't necessarily mean that your site is the healthiest for water quality or habitat quality. Sorry. Okay. So our program follows, or it has an EPA quality assurance project plan. And this is just done to make sure that all of our volunteers undergo a QA/QC, a quality assurance quality control training before they actually start monitoring and taking samples. So what this means is that only individuals are certified. We do not certify whole groups, just the individuals. And this is just to ensure that everyone is following the same protocol when they are taking their samples. And it just makes the data more reliable. An individual certification is valid for 1 year. And then after that year, you can attend a recertification workshop. Volunteers must attend that each year, and only certified volunteers can submit data to the Adopt-A-Stream website.
Thanks, Jackie, for explaining all those things to us and getting us kind of kicked off with our workshop here today. So like Jackie is saying, we do have that QAP, that Quality Assurance Project Plan, and that is kind of what these workshops that we provide, you know, are a part of that, just like Jackie is saying, that we need to all be kind of on the same page as far as protocols, what exactly we need to be doing out in the streams, how exactly we need to be documenting things. And that might sound a little overwhelming, but it's definitely not. Once you get your first 1 or 2 under your belt, you'll be a pro, we promise. But in order to become a certified volunteer, there are a few steps to that. And one of those, the first thing is just getting started with a workshop. And this is kind of the first part of that for all of y'all that are here with us today. We're going to provide you with some background information and get you pretty much everything you need to know to pass our certification test. That's the written evaluation. We like to say quiz because test can sound intimidating, because it's definitely not too bad, but there are some things, like we were saying, we just want you to be aware of, and that we're all on the same page. So I don't think we've mentioned it yet, but anything that's in bold in this PowerPoint is something you'll probably want to remember for when you take the test. And of course, you can come back and watch this video again. It'll be posted online. And also, this PowerPoint is also posted on our website as well, so you can review it at any time. But this will have all the info you need to take that test, and then you'll also be responsible for connecting either with us at the state office or with your local coordinator, which we can facilitate at the end. But we do have trainers kind of across the state that can help you get some practice in the field, social distance style, and do this kind of field portions that we're going to be talking about later in the presentation. So that's going to be part of becoming a certified volunteer, and then also identifying 20 macroinvertebrates with at least 90% accuracy. So we just have some vials that you guys will look at if you end up taking this certification quiz, that you'll need to be able to look at the bugs and identify what they are with the help of a key and with some practice beforehand. So. Thanks, Jackie. All right, so before we get started, we like to talk about this in every single workshop. We think that this is incredibly important and valuable information for us all to be aware of. So if I were with all of you in person, I would ask you all a question and hope that you would respond, or do something where we all are cupping our hands like this and say, what is a watershed? Well, a watershed can be demonstrated like this. If we cup our hands, we can see that we have some high boundaries here, kind of being these where my thumbs are, that kind of flow down into the cupped part of my hand where this crease is meeting. That's the lowest point, and that's the one common point in this whole area of my hand where everything meets. So that's exactly what a watershed is. It's all of this area of my hand and then where everything will run over and drain to a common point. So as we can see here, the definition, a watershed is the land area. Important to note it's the land area over which all the water and things are flowing, because of course water is a very important part of a watershed, but everything the water touches as it runs over the land becomes incorporated into those water bodies. So it's just important to remember that, and especially being in the non-point source kind of realm where we're thinking about things maybe just litter in the streets or like urban runoff or agricultural runoff. Those are all things that we experience and we can see our watersheds being affected by. So yeah, if you don't know what watershed you're in, we encourage you to find that out. You can look on this map. This is the 52 sub-watersheds of Georgia. We have 14 major ones that are, bigger, kind of combined. But also a watershed can be as small as your backyard watershed, the creek in your backyard. So there's quite a range, but they're all important. So our next slide is going to start talking about where, when, and how often. And we're kind of jumping back again to talking about macros. We took a little detour there to talk about the importance of watersheds, but now we're going to kind of focus in on macroinvertebrate monitoring. So we're wondering where, when, and how often. So how much time is this going to take from you? Where do you have to go to do all of these things that we're going to be telling you about today? Well, where, if you already have a site location, if you already monitor with our program, or maybe you have a Rivers Alive site that you clean up at, or yeah, anything like that, or you don't have a site yet, that's totally fine, but you have a place maybe in your backyard or in your neighborhood or in your grandparents' neighborhood that you could be interested in finding out more information about. You can just pick a site and that would be where you would want to go. And we want to go to the same spot every time when we're doing this macro monitoring, and the same with our other types of monitoring as well. So when do we want to sample? Ideally, we want to sample at the same time of day. And that's important because temperature can change and that can have an effect on what we're finding. But also during normal flow conditions. This is going to be pretty important when we're talking about macros. We don't want there to be flood conditions or really strong currents that are abnormal in your stream, because then if it's not normal for your stream or your other kind of water body that's not a stream, then those macros could be washed away. So we want to make sure it's normal conditions around the same time of day, and we want to make sure that you dedicate a pretty good amount of time to doing this. So typically 1 to 2 hours is about how long this whole process will take. Once you're pretty good at it, it could take a little bit longer the first couple times, just because we're getting used to being familiar with the different kinds of macros that we're seeing. And then how often? So this is going to be quarterly sampling, so once every 3 months that we're going to be doing the macroinvertebrate monitoring. Let's see. Yeah, and then our next slide is just kind of talking about those safety considerations. So I already mentioned flooding, flood conditions. We don't want to go out in that, not only because we might not find bugs, but primarily because we want you to be safe. And we don't, that's our top concern. We don't want anyone to get hurt or anything like that while they're out in the field. So if you ever feel uncomfortable about going out, your safety is definitely more important than getting and collecting a sample. So make sure, there's no storms while you're going out, which may be hard since it's been storming nearly every day here in Georgia. But never sample when it's storming. And then if you can go with someone else, try never to sample alone. Use protective equipment. So if we were to alter this slide right now, we'd probably say wear a mask if you're going out with someone else. We do have some COVID safety considerations posted on our website if you're interested in and checking those out. And then always, of course, check for permission if you're going on private land. And this is not a comprehensive list, but we just want you to feel comfortable and safe when you're out monitoring.
All right. So diving now deeper into the topic of our workshop today, what are macroinvertebrates? So that's a pretty big word, but if you just divide it into two, you'll figure out it's not too bad. So invertebrates, if we take that later part of the word, invertebrates are just organisms that lack a backbone. So we are vertebrates, we have a spine. These critters do not have a spine. And if we look at the first part of the word, macro just means large enough that it could be seen with the naked eye. We don't need to look under a microscope to be able to see these little creatures. They are large enough that you'll be able to pick them out of the sediment, pick them out of your samples, and be able to identify them. So macroinvertebrates. The organisms that we sample for, these macros, are benthic. So that just means that they live at the bottom of the water body, and depending on the type of macro, they may be within the substrate, between rocks, but basically they just dwell at the bottom. Macros live in all different types of stream habitats, and they derive their oxygen from water. And that is really important, really key, hint why it's in bold. But yeah, they derive their oxygen from the water. So because of that, they are easily impacted and influenced by the quality of the stream and the quality of their environment. So stresses that occur to their environment, it can be naturally occurring stresses or man-made, man-induced stresses. All of this will impact the macro populations. There we go, finally it's decided to change. So, like I said, they are easily influenced by the stresses in their environment. So because of that, they are really good indicators of water quality. Other reasons why they are, used as these bioindicators is because they're not very mobile. So these macros, they're not going to run away from you very quickly. They're not going to dart away quickly like a fish. They're pretty, docile and easy to catch relatively. They will be present in all stream events. And however, recent heavy rains can affect your results. So kind of like how Bailey mentioned before, if it's a flooding event or high currents, you don't want to go sampling. For one, that's a safety hazard. But two, if you're going out during a strong storm event or during a high current, it can actually, just wash these macros downstream and they're no longer at your site. So that's why you want to make sure that you sample for them during normal stream conditions. They're big enough to view and identify easily. They are affected by the physical, chemical, biological conditions of the stream. And just keep in mind though that the values of these populations may differ depending on where in Georgia your site is. So if we're looking at a stream, we have different habitats that can be found within this one stream. So vegetative margins are usually your areas along the edge of the stream. And this has a lot of overhanging vegetation, dropping off a lot of leaf litter down into the stream and introducing that into the system. You also have your substrate. So kind of like how I said before, these macros are benthic. They live at the bottom within the substrate. So this could be sand, rock, or gravel. That is what we call coarse substrate. So those are, larger in size. And you know, when you have these collection of rocks or gravel, it can create riffles, which are shallow areas of the stream, and the water will flow pretty rapidly. You know, and it'll kind of have this, you know, bubbling, like almost like water rushing effect over it. And then you will also have organic matter, which can be composed of leaf packs. So like I said, the vegetative margins, they're introducing those leaves into the stream. Well, leaf packs are decomposing. So that can be like the decomposing leaves or just decomposing vegetation that is actually submerged in the stream itself. Also, woody debris, again, just decomposing trees, roots, branches that are submerged in the water and decaying. So these are your three main habitat types found within a stream. And this photo, here we tried to, you know, really capture all of these different types of stream habitats. So I'm not sure if you can see my mouse or not, but I mean, you can see, okay, great. So you can see, you know, we'll have some vegetative margin, like the areas where there's vegetation, you know, if this was, if this had more plants growing around it, like the riparian zone, then, you know, it would be, this whole area would be heavily influenced by it. But we can see some riffles here, and, you know, where the areas where there's some rocks. So when that water is really flowing, it will be flowing quickly over the rocks. And then you can see we have some woody debris, some roots that are probably just decomposing over time in the stream itself. So you can find all of these different habitat types in one stream, and that's important to note, especially when you start sampling.
Thanks, Jackie. Okay, there it goes. Sorry, I was like, I think there's a lag. Okay, we're good now. Yeah, so Jackie just told us we can have several different types of habitats just within one type of stream. But even going a little bit further than that, there are different types of streams that you can find within the state of Georgia, and really beyond, really anywhere you are. So, but specifically, if we're talking about Georgia, we're going to be looking at what we classify as rocky bottom streams, and then we also have muddy bottom streams. So we do have two different types of protocol that we go into when we talk about these kind of things, or these different types of streams, which I'll go into detail a little bit more about. But just to give you some background information, rocky bottom streams typically found in North Georgia, the Piedmont region, and as you can tell by the name, rocky bottom, we typically see rocks, pebbles, and more bulky, kind of coarse substrate. So that's, you know, the bottom of the stream bed. If you're up in North Georgia, where you see headwater streams, you could see big boulders, things like that. But it also can be, in that previous picture that Jackie showed us, those small kind of rocks that were there in the middle of the stream, that could also be classified as rocky bottom. So looking at that substrate is really important. It can tell you a lot about the type of stream that you're looking at, but also you can kind of know by a few different characteristics. So rocky bottom streams typically have faster moving water. Faster moving usually also means colder water, and they're going to have more riffles, because if you have more of those rocks or boulders, like Jackie was saying, when you have those collections of rocks and water's flowing over them, you're naturally going to be creating turbulence or bubbles in the stream as water is running over those rocks. So you'll see riffles, and you'll see, so you'll have like a riffle area, then you may have a run, which is just a straight area of flowing water, and then the natural sinuosity of the stream, you'll have some pools as well. So riffle, bubbling, a run, water just flowing kind of straight, and then at the bend, you may see a natural pool at the curve where water may not be flowing as strong as it could be, you know, in that run area. So there are some great visuals of this, which would do probably a better job just looking at it than I could be explaining it. So we can share those with you as well. But our other type of stream is a muddy bottom stream, and those are typically going to be found in South Georgia, where the geology is quite different than it is here in North Georgia. So North Georgia, if I was in Atlanta right now, if I was at my office, I could see Stone Mountain from my office building. So we know that North Georgia and the Piedmont region are heavily influenced by granite. We naturally have that rocky geology, but in South Georgia, what's written in all the songs is we have that red Georgia clay, and we have kind of on the west side of Georgia we have limestone, and that's a little bit different, and we're going to see how those things are going to be showing up in our different types of streams. So South Georgia, where we'll find muddy bottom streams naturally, and then urban environments is where we could find some muddy bottom streams unnaturally. That's going to be influenced by man and different types of maybe construction, where you're getting that erosion and sediment flowing from maybe construction projects and coating that stream bed and making it look like a muddy bottom stream. So also we're going to have slower moving water here. We're not going to have those rocks, so really not as many riffles or anything like that. We are going to have fine sediment on the bottom of that stream bed. So if you are in an urban area and you're looking and you say, well, this kind of looks like a rocky bottom, but there is some really, really fine sediment, we just want you to take a look at your whole site area where you're going to be sampling and choose the type of stream and that methodology that's going to be most representative. So that might not make a lot of sense when I'm saying it, but when we're seeing the different types of habitats and you're actually looking at the sediment, it'll make a lot more sense. But if you have any questions, feel free to drop those in or ask us at the end as well. So our next slide is going to start telling us about the rocky bottom sampling method. So like I said before, we do have two different types of sampling methods for each different type of stream. So our first one, rocky bottom methods, we can see in this picture here, this net that they're holding up in this photo is a kick seine. And so that's what we're going to use to sample the substrate of this type of stream. So when we're sampling here in a rocky bottom stream, we want to take 3 different substrate samples. And what we're going to do when we're taking a substrate sample, we're sampling that for those benthic macroinvertebrates that are living there in the bottom or the substrate of the stream. So what we're going to do is just like in this photo here, we're going to have maybe 1, but like they have 2 here in this photo, people holding this net kind of anchored down into the stream. And we are going to have someone standing in front and do what we call the famous macro shuffle and right in front of that net. So I wish we could do a demonstration for you today because it's very fun. But that's what's going to give us all of the sample that we need into our nets. And how that's done is we have the stream, the net in the stream anchored there by some rocks. Maybe we're seeing some big rocks or smaller rocks that we can pick up and brush off and kind of anchor the bottom of that net down into the substrate. Because if we're kicking in the substrate and kind of trying to disturb those macros that are living in the rocks and in that coarse area, we're kicking all of them up, and we don't want there to be a gap between the substrate and the net. So we want the net to be anchored down in, and then our macro shuffler is going to be in the front, kicking around, disturbing that substrate, and making it look really murky, and kicking all those things so that they'll flow right into the net. So another important thing to note here is that we're facing upstream. So the water will be flowing this way. We're going to have a macro shuffler here, and the water's just going to be flowing right into our net. We don't want the water to be flowing the opposite way because then we won't catch anything. So we need to be mindful of that. Then once we do that, you're going to do it 2 more times. We want to get 3 substrate samples. We just want to make sure that we are getting representative areas. We'll make sure that we're looking in places where we're actually going to find macros. So for rocky bottom methods, we usually say look for riffles and try to sample maybe 2 to 4 feet below a riffle so that you are able to get in that area, riffles are going to have a higher dissolved oxygen level, because if we remember, like Jackie told us a few slides ago, that these macros derive their oxygen from the water. And if we're in a more highly oxygenated area, we're going to be likely to find more in this rocky bottom habitat. So we're going to take, 3 different samples, and they don't all need to be in that first place that you did it. We actually encourage that you go to 2 different locations in the stream and get those substrate samples. So after you've done that, you've got your samples, you're going to probably put them in some buckets, rinse your nets with water, and make sure that all of your macros are in your area where you can kind of sort through and look through and then identify, which we're going to be talking about later. We're also going to take 4 organic matter samples. So you notice the 3 substrate and the 4 organic matter are in bold. Those are definitely things we want to remember during this methodology. So we've already got our 3 substrate. We're going to take 4 organic matter samples. And if you remember, like Jackie told us, organic matter is going to be submerged and decaying materials. So, or vegetation, excuse me. So we want to make sure that we're not getting some freshly fallen green or yellow or red leaves, even though they're pretty. We want to get the yucky brown, slimy stuff. That's where we're really going to find all of the great macros living. They're kind of feeding on that decaying vegetation. So we want to grab that. And we say just estimate, but about 1 square foot. So a big handful if you can. And if you can't get 1 big handful that size, just kind of estimate and add as best you can. So our next slide is going to tell us, about muddy bottom sampling. And this is a little bit different because we're actually going to be sampling 3 different habitats, whereas before we were only sampling substrate and organic matter. We're going to add vegetative margins in the muddy bottom sampling method. And this is because of the finer sediment that we have in muddy bottom streams is not always as conducive to macros and what they like to survive in or like to live in. They like to kind of find areas that are a little bit different than fine sediment, at least sometimes. So this is another thing that Jackie mentioned earlier. Just because you get a value that may be lower or that might just be different than what you would get in North Georgia, that doesn't mean that it's bad or your muddy bottom stream is unhealthy. We're just looking at different habitats. And anytime there's a different habitat, there's going to be different organisms that thrive there. So that was a little soapbox, but so what we're going to do now with our sampling, our muddy bottom sampling, is use a D-frame net. So you can see in this photo here, the net that this woman is holding up is shaped kind of like a D at the top, and that's what we're going to be using. So the first thing we'll do, or I guess you can do these really in any order that you like, but for our purposes, it kind of makes a little more sense if we start outwards and work our way in. So Jackie told us about vegetative margins. Those are at the edges of the stream where we're going to see that overhanging vegetation. We're going to see some roots living in the water and making their way, getting some nutrients in there. We want to sample at those vegetative margins and look for macros that really like to use those roots and those overhanging vegetations to thrive. Especially, I know dragonflies really like to live on roots. So if you're ever in a river and you get over near the edge and you see some little exoskeletons, those are probably dragonflies, which we can show you some pictures of soon. So those are going to be those overhanging roots, the overhanging vegetation and the roots that are in the water. We're going to take 7 samples in that area. We're going to move on to organic matter. 4, same as in the rocky bottom, 4 organic matter samples. This is primarily going to be woody debris. So in muddy bottom streams, we don't typically find as many leaf packs, but you usually find more woody debris. So maybe that's a big tree or log has fallen into the river or stream that you're sampling, or maybe just some big branches and different things like that. So we can rub those just like we were rubbing those rocks in the rocky bottom sampling. We're going to kind of do the same here with this woody debris and get 4 samples from that organic matter, which again is submerged and decaying. And then our last sampling type that we're going to do here is the substrate sampling. So again, we're taking 3 substrate samples, and that's going to be in the coarsest area you can find. So sometimes in a muddy bottom stream, you're not going to find anything different than just silty sand, and that's okay. But if you can find an area where you find a little more pebbles or gravel, definitely target those areas when you are sampling for substrate. And we can talk about, if anyone has a muddy bottom stream that they're sampling here and they have any questions, we can talk about, oh, what is it called that Harold always does? The thing where you stir the, what is that called? Of course now I can't think of it right when
Elutriation.
Elutriation, thank you, Jenna. Yeah, so elutriation is a really cool technique when you're taking substrate samples in a muddy bottom stream or in an area where you have really fine sediment, because we're sampling that fine sediment and that's all going to get stuck in our net. It's not going to wash right out with the water. So if you take a sample and then you have just a big, pile of mud or sand, that's going to be really hard to sort through and take a long time. So there's, I have to send that video or try to post it somewhere. I think it's on our Instagram page, but a process called elutriation where you can kind of sort out that substrate and pull the macros out from there. So we can share some more information about that. All right, our next slide. This is something that is really important for our purposes, but also to a lot of other scientists and things as well. So macroinvertebrates are really, really amazing creatures. They can tell us so much about water quality and habitat quality, and a lot of those reasons, why Jackie was talking about earlier, why we use them, they're indicators. And the reason that they're indicators is because each type of organism that at least we have in our field guide, which is on the next slide, so you'll get to see a visual of that. Each of these organisms has a known tolerance value. So really what that means is that they can be put into these categories. We know how sensitive each type of organism is to pollution. So we have pollution sensitive organisms where you need really great water quality. They can't really handle a lot of pollution. They're, of course, as it says, very sensitive to that. They need higher levels of dissolved oxygen, so you're only going to find them in high quality streams versus somewhat pollution tolerant and then pollution tolerant organisms. Pollution tolerant organisms, you're really going to find pretty much anywhere a healthy stream, and then a not very healthy stream as well. They can really make it anywhere. And then that somewhat pollution tolerant falls right there in the middle. But these macros are really important for that reason. We know what they're sensitive to, and we can say, we know that this must be a pretty healthy stream if we're finding these types of organisms there.
Yeah, and before I transition to the next slide, a point that I had made earlier was, we are looking at abundance and diversity, but we really focus on the diversity of macros that are found because, abundance, just because you find a high number of macros doesn't mean those are I don't want to say the good macros because, they're all important, but if you find a high number of pollutant tolerant macros, then that might mean that your water quality is not too good, because they can survive in these low levels of oxygen and they can take the pollution. So it's really about, again, the diversity of macros that we find. Okay. So I'm just going to start to introduce you to some of the forms that we have and the resources when you start to sample and input your data to the website. So this is a screenshot of our field guide. It is found on our website, and I can attach a link to that here at the end of this presentation. But this is just a brief overview of the different types of macros we sample for. There's a really helpful ruler on the side of the field guide that can help you determine the length, because a few of these, we're collecting them when they are in their larval form, not when they're in the adult form. So they can look very similar. Here, you'll learn about the different characteristics as you go through your training, but there's different characteristics here that help determine or differentiate between each type. Not only is size important, but different characteristics, the number of tails that they have, if they have, how many appendages they have, if they have an armor around them or what. So this really just goes into more detail of the things we are looking for when it actually comes to identifying the macros. So, excuse me, on our sampling form, we always ask for you to take just a visual observational data of your site. And if you have a site and you go do chemical sampling or bacterial, you only need to fill this out the one time that you're going out to sample. So if you have a chemical form, this section will also be attached at the beginning there. Just know that you only have to take the observations for that one sampling event. But we go over different details like recording the time that you spent sampling, the weather conditions, the amount of rain, the water levels or the flow, the odor of the water, surface color, clarity. All of these are really important observations to take, and because they can also tell you about the data that you're collecting. If you're going out, maybe the flow is lower than normal, maybe that will also show in the sample of macros that you just collected. So it all intertwines with one another, and all of this can be included or entered onto the website. So this just goes into more detail of the observations that we ask for you to take. I will note here that it asks for photos. However, on our submission form online, there isn't a way that you can take that you can submit your photos, but photos are still really, really important to take just for your records and for your own documentation. So we still encourage that, and we are working right now to develop a system for our volunteers to enter and submit their photos. You can also take note of any trash that you find at your site. That's really important. If you notice that there's more trash than usual, maybe you want to organize a cleanup at your site. We have our sister program, Rivers Alive, which focuses on waterway cleanups, and we can help you start a cleanup, provide you some materials to help you accomplish that. So this is the actual macroinvertebrate form, and I find it easier to go through when you're actually there at the site once you've collected your samples, but I can do just a brief overview of what it includes. At the top, you take note of the type of stream that you're at, if you're at a rocky bottom or the muddy bottom stream. We want you to make note of the method that you use, the type of habitats that you sampled, and here we have written directions for how you record your data. So here we have our taxa groups of the different macros, divided into those three categories that Bailey had mentioned: the pollution sensitive, somewhat sensitive, and then the pollutant tolerant macros. So when you take your sample and you start to separate them out, you're going to separate them by the type of macro that they are. So, okay, maybe you find 5 stoneflies and 2 gilled snails and 3 water pennies. So you'll want to make note of the abundance, so the total number of those that you found. And we have an abundance code to make it easier for you, and that's here in the directions. Rare is if you find 1 to 9 of those macros. Common is 10 to 99, and dominant is over 100 individuals. So you'll make that code here in the box, and you'll do that for each group if you find the individuals for each one. And then you come down the form and you're going to take note of the number of taxa groups, and then multiply that number by 3. So like I said before, if we found stoneflies, water pennies, and then snails, gilt snails, well, that is 3 different groups. So that would be the number that you reflect there, and multiply it by 3. You continue that for the other groups, add those together as it says here in the directions, and that will tell you your water quality rating. So it's a lot, and I'm a very visual and kinetic learner, so this took me a while to get used to. Once I was actually out in the field, I think it makes a lot more sense once you're actually out there counting and seeing, okay, this is what abundance is and diversity, and these are the groups that we're looking at. And then we also have other groups that you may find in your samples, and if you find them, that's great, and we'd like to know about that. Some Asian clams, like some invasive species, if you're able to identify them, you can make note of that. Tadpoles, those are all at the bottom though, and they don't influence your water quality score, but they're just good to know.
Absolutely. Thank you, Jackie. Yeah, and that was a great explanation. It is kind of difficult, especially because we haven't shown you any macros yet. And you may be familiar with them already, but if you're not, then you can be like, I don't even know what a stonefly is. But once you are looking around in your stream and you're familiar with them, you get some practice, and you have that field guide that Jackie showed us, and you're able to use that to help you identify. It's going to be a lot easier filling out that form. And I think that Jackie did a great job explaining, we don't want to count. Let's say that's the reason we have diversity codes. We don't want to put that we saw stoneflies 10 different times and count that in our calculation, because that's going to be a little bit different. That factors in abundance, and we're not trying to do that. We're just looking at diversity, like Jackie said. So that was great. I will say that once you're putting it in online, it does the calculations for you. Not that it's hard, but it is kind of helpful to just see that all laid out for you. So if you just want to make note of what you found and then put it in on our online data forms, which I'm about to talk to you about, it might be a little bit easier. So yeah, we do suggest that you take a paper data form, because our online data forms are not super mobile friendly. Tried it a few times and it's not really the best. Something we're working on, we've been working on for a while, but we don't have anything ready for that yet. So definitely take out a paper data form when you go, make notes of what you're finding. But then once you get back home, please, you'll have access, once you're a certified volunteer, to submit the data that you're finding to the online database. So that's accessible at upstream.georgia.gov. You can go on there and look at other people's data right now if you're not certified, but you'll only be able to enter data once you're certified. So as soon as possible, check that out after you've collected your data on your data sheet. And then I think there's some slides on here that we're going to kind of show you what that online data form looks like. So this is showing our main home page, and you see there's a data entry tab, and if you click that little caret or drop down, it'll take you to the data submission page. If you click on that, it's going to bring you here, and we're working on making this a little bit more easy on the eyes and a little easier to use as well. So hopefully we'll have those updates ready for you soon. But as it stands now, this is the data form. online, and it pretty much mirrors that paper data form. So you should just be able to follow along and click, click, click and put things in. So if you filled out that paper one, you should just be able to copy that right over in here. So we have the weather and observations first. So you can see the tabs at the top of this screenshot here where it says site. This is the site page. So we're going to be putting in all of the observations that Jackie talked to us about how important those are. Those are going to be on this data form. So you'll fill that out first, putting in all those observations, and you'll click submit. Then you'll get a pop-up saying, thanks for submitting this. And then you can go to the next tab, which will be, in this case, macroinvertebrates. So click on that tab, and then again, it's just going to mirror the macroinvertebrate form. So you can just copy all of that information over right here, and then click submit again. So you'll click submit after every single page that you fill out. So if you're doing also chemical and bacterial, those are done monthly. So you might just tack on macroinvertebrate, that's once every 3 months. So you'll just be able to add all of those in at the same time if you end up doing 3 in one day, which if you do, amazing, great, good for you. And you can just do them all at once. But make sure you're hitting submit after every page.
All right, so this is just, a quick little graph just to show you the different ways that volunteer data can be used. So for, a lot of times it's used for educational purposes. We have a lot of school groups that will go out and monitor and incorporate this different protocol into their curriculum. But a lot of times it's used to establish that baseline water quality conditions. And when there are, big changes in those conditions, then volunteers can notify their local water department, whoever is the water authority or the governing authority, they can notify them of the observations that they have seen, and now they have this data that backs up their observations, and they can present that and then work to figure out what's the source of this and what's causing this to happen. But as you can see, just volunteer data can be used for a whole variety of purposes: research, advocacy, or just watershed planning. It really, the uses are very diverse, just like our volunteer groups. These are some helpful links to help you on your monitoring journey. So this link is to find your local coordinator, and I will include these all in the chat afterwards for you to access them. But the local coordinator, this is the trainer in your area who could help you maybe find a site, help you find a group to get involved with, equip you with monitoring equipment. We encourage our volunteers to establish that relationship with their local coordinator. And also, if you're using this presentation as part of your certification, this will count as the classroom portion. And we encourage you to contact your local coordinator to complete the field and identification portion of this course. We also have our Getting Started page on our website with a lot of great resources to point you in the right direction and to get you started. Volunteer FAQs to answer all those questions you may have. Of course, if your question's not listed on there, and or you just want to talk to us, feel free, just to send us an email, and we are more than happy to, get you pointed in the right direction, help you with the training, get you the equipment you need, whatever it may be, just give us an email. I also included a link to our additional resources page of our website. So on this page, we have many different resources for all of our different monitoring programs. So for macro identification, we have many different links, just quizzes or online games that you can use to help you get those identifications down. So I encourage you, especially as you're learning, to check those resources out, and that will make identification a whole lot easier. This is just a screenshot of the local coordinator page. You can look it up by your region, your service area, and your coordinator, their contact information will be listed on there along with the organization. But if you don't see a local coordinator for your area, just send us an email and we can send out our own staff to help with the training or to get you started. Before I do this little slide real quick, I just do want to mention that these PowerPoints are available online, and at the end we also have, I don't even know how many slides it is of macro identification. So this is where we divide them up again into those sensitivity groups and provide the field guide picture blown up for you with those characteristics. Some of the ones in bold are those defining ones that really will help you identify them. But we provide these for all of the macros. We identify all three groups. But that's just here at the PowerPoint. I just wanted to make note of that.
I will say really quickly, for the macro ID portion, those things that are in bold, you don't have to remember every single one of those things for the test. I just kind of realized that. I had never really thought about that before. But these are just really to help you. So starting at macro ID and down, things that are bold, that's just something to kind of get you started, like Jackie's saying, and giving you those prominent characteristics, not something you need to have memorized to, you know, spit back out for the test. So just something to keep in mind.
That's a really good point. Forgot to mention that. Yes. Any questions? Feel free to drop your questions into the Q&A. And
yeah, there were a few questions or kind of comments and questions. Chelsea Thomas asked, I believe she's talking about tadpoles, and what if you see them but they're not in your sample? So if they're just around the area where you're sampling, would you include them, would you not?
Yeah, you can definitely just say that bottom part of the form. So we'll have, you know, these different types of organisms laid out that we're not necessarily counting in our macro sample. Like you're saying, tadpoles are in there. So you could definitely put, you know, usually when you see tadpoles, you're going to see a lot of them. So you could just, you know, kind of give us a rough estimate. And then we do have a comment box at the bottom of every single data form. So I definitely would encourage really anyone on any type of monitoring to take advantage of that comment box, because that gives us a lot of context. We do get emails every time a data form is submitted, so it can give us context as far as, you know, what you're seeing in your stream 200 miles away while we're in the Atlanta office. So yeah, if you see them, you know, 200 yards downstream while you're just hiking to your site or something like that, yeah, feel free to just let us know.
Thanks, Bailey. Another question from Gretchen. She's asking, the stream bottoms in her area are very sandy. Does that matter?
Yeah, so it's going to matter as far as determining what type of methodology you're using when you are sampling. So if you do have that fine sediment where you are, Gretchen, yeah, you're probably going to want to choose that muddy bottom sampling method and using that D-frame net and sampling as many habitats as you can. And I did notice you put in the chat Blackwater rivers, just because they have a low DO doesn't mean they're polluted. You're absolutely right. And that's naturally occurring. It's a slower moving river. It has more tannins. You know, these are all things that happen naturally. It's not polluted. but just because the habitats are different for different macros, you know, that could kind of show, again, that difference in North and South Georgia values in streams. So no, it's not saying that your stream is polluted, but it could just mean the habitats that are there might not be as naturally conducive to macros. So.
Awesome. And Johanna's asking, will we get certified for both rocky and muddy sites at the same time, or only for our local region?
So this, when you get certified, you can do either. Am I correct with that, Bailey? Is that safe to say? Okay.
Yeah, absolutely.
Yeah, so you are learning about both types. And of course, the type that you use depends on your sampling site. But when it does come time for that recertification every year, you know, you will go through the same course. So, you know, it's kind of expected that you would know the, you know, rocky bottom and the muddy bottom protocols.
Yeah, absolutely. Just like Jackie's saying, when you're taking this test, you're going to be tested on both types of protocol. Just depending on where you are, you could be in areas where there could be both types of streams. So we would like you to know both. So yeah.
I have a question just for people who this may be their first time attending, let's say, an adaptive stream workshop. So do they need to do anything to prepare if they wanted to meet their local coordinator for the field portion in the test? What would you suggest?
Great question, Jenna. I can start, and then Jackie, feel free to add anything because I'm sure I'll probably miss something. But yeah, I would say especially for macro, before we do a workshop that's in person where we're going out and trying to find bugs and we're going to show you bugs and vials, even if you are familiar with maybe one or two, we suggest going through and checking through those resources that Jackie mentioned. We have some games, we have those, yeah, and Jackie just put the links in the chat. So definitely would encourage you to check out the identification resources that we have posted, because Harold, who is our manager and has been with the program for many, many years, usually says if you're not familiar with at least 4 to 5, chances are you're going to have a difficult time on the identification portion. Because there are so many macros, we're really lucky in our state that we do usually find a lot of different kinds. That is more for us to remember and for us to be familiar with. So definitely check those out. But talking with your local coordinator, all of our local coordinators are amazing and they're definitely more than willing to help you. So as far as talking with them, I would just say if you don't have a site picked out yet, you know, come to them with questions about that. And if you do, then they can usually equip you with the materials and equipment that you need. And if not, that's definitely something that we can help with. So that kind of might be a little scatterbrained. But that's just kind of an all-encompassing thing for workshops. Jackie, do you have anything to add?
Yeah, I also just included the link to our workshop presentation page. So I would recommend, if you're going to follow up with your local coordinator after this, to go ahead and take a look at this PowerPoint, because it is posted on the website and you'll be able to access it. And I know what really helped me is using this PowerPoint as a study guide. So kind of like how Bailey said before, everything that is in bold is something to note for your quiz. So if you're going to go meet your coordinator and you know you're planning on taking the quiz, not only are those additional resources really helpful, but this presentation, this PowerPoint itself, could be used as a really helpful study guide.
Yeah, thanks, Jackie. And definitely we don't want to intimidate you and saying that you have to study so hard for this quiz or anything like that. Yeah, no, and I know that's not what you're saying at all, but it is a really helpful resource. You know, we are teaching you what you need to know, teaching you what is on the test, and, you know, just trying to equip you to go out and monitor and go out and learn, you know, what are macros and how can I find them and what does that tell me? that's really all we're trying to get across here today, and with the test as well. So nothing to be super worried about, just information that we would like you to be familiar with.